the end, we might have few minutes for some questions. I'll try to be fast and not to bother you again with the same details about the ticks. However, I still keep some of the stuff from the first tick talk, maybe for those that were not here last week, and also to remind you about some direct links between ticks and the parasites they transmit. Okay, so in last weeks we discussed that among the ticks that are present on domestic carnivores in Europe, there are basically two dominant and one almost dominant species. The dominant ones are Ixodes ricinus in, I would say, Central Europe, from the north up to the Alps or southern slopes of Alps, and Rhipicephalus sanguineus, which is in fact a complex of related species, which is common on carnivores more in Mediterranean region of Europe. And there, Dermacentor reticulatus, kind of in a band throughout Europe, and it's a species which is quite fast broadening its distribution range. We discussed that the populations of ticks, their abundance, their presence in environment and on dogs strongly depends on the season, but also on the latitude and altitude. Not surprisingly, the ticks in more Mediterranean parts of Europe, of course, including Sardinia, are emerging much earlier than the ticks in Scandinavia and vice versa. And also then the season for the transmission of the diseases, it's quite differing between different tick species. Yeah, so this is just some background for the diseases they do transmit. This is a picture that we visited yesterday, clearly showing the difference between two important canine Babesia parasites and their distribution. Babesia canis associated with Dermacentor reticulatus, the orange dots in the map on the left side, mainly in the central part of Europe. Babesia vogeli associated with brown dog tip of the genus Rhipicephalus, the green dots on the map. So mainly Mediterranean part of Europe, parts of North Africa, Turkey, and including, of course, Sardinia. It's a dominant tick that you definitely all of you know from your dogs and possibly also cats and some other animals. We also discussed already that these three tick species clearly differ in their vectorial competence for different tick-borne diseases. And you might remember that we also associated that with the life cycles of the ticks and with the way they change their host in the population during their life cycle, with quite opportunistic feeder, which is Ixodes ricinus on the left side, transmitting also to dogs and cats, of course, the diseases with broader spectrum of the hosts like anaplasmosis, borreliosis, Babesia divergens, and so on. And on the opposite side of the spectrum, Rhipicephalus sanguineus group of ticks, here on the right side, transmitting quite host-specific pathogens of canids, including domestic dogs, of course, in Europe, dominated by Ehrlichia canis, which you probably know. It's the pathogen that you definitely should know because it's present all around Mediterranean, causing symptomatic infections on dogs. Anaplasma platis, responsible maybe for more of asymptomatic infections. Rickettsia conori, important because it's transmissible to humans as well. And Babesia vogeli that we already discussed before, plus Babesia gibsoni, which is more common in the tropics, but occurring also in the Mediterranean. And then Cercopithecilaria, the nematodes, the skin nematodes that we will discuss at the end of our talk. So if we look to a little bit now more closely to the protozoans that are causing the diseases, we stop speaking about the ticks and we truly focus on these pathogens, we can conclude that there are several. They commonly overlap in their hosts, and through the ticks, they are commonly shared between free-ranging carnivores and the domestic carnivores. And that's true for all of them with smaller or bigger extent. In general, tick-borne diseases in dogs and cats, it's quite a dynamic field. It's area of different interests, and of course it's a topic which is extremely important for veterinary practitioners, because all dogs and significant part of the cats can be or are infected by ticks, and the animals infected by tick-borne diseases will represent a significant part of spectrum of your patients and clients in the future. So what's dynamic in this area? Why we maybe speak more about the tick-borne diseases than before? For sure, there are several things that stays behind. Definitely, these are the new diagnostic tools. With development of mainly molecular tools for the detection of pathogens, we are more and more understanding the distribution. We are more often and more frequently detecting these pathogens in dogs and cats, and we are also able to identify new species, either new species new to science, or at least species new to given geographic areas. Then, as we already mentioned repeatedly, there are also quite fast changes in the distribution of the vectors. The ticks are moving together with traveling animals. The ticks are moving together with migratory birds. Ticks are establishing new populations. And we also should consider, and I do not mention it here on the list of points, that also the lifestyle of people and animals is changing. Yeah, we have more time than a few decades ago. We travel more frequently with our animals than a few decades ago. We go more often with our dogs to the nature, or I hope also you do, than a couple of years ago. And of course, this is making quite intense interface between the environment as animals living in the environment as reservoirs of tick-borne diseases and our dogs and cats. Yeah, and in some of them, what also making this field quite complex or complicated is that we already see the emergence of resistance to some treatment. Typical example is Babesia gibsoni. I will speak about that a bit later. And tick-borne diseases, similar to other vector-borne diseases of domestic dogs or cats, are also associated with a process that we in Europe called rewildering, where, and we discussed that one last week also, when we kind of allow the free-ranging carnivores to live closer and closer to us, and it also intensifies the transmission of the diseases between them. And on top of that, in Europe, we see the new mammal species coming that are hosts of the ticks and can be hosts also of the tick-borne diseases, like raccoons across Europe. And all this is, I would say, giving even more speed to the development, because more we work on these animals, of course, more we see, more we know about these diseases, more topics to study are emerging. However, and this I want to stress, there is no evidence that there are more and more tick-borne diseases seen in dogs and cats. I think it stays mainly with the diagnostics tool. So the tick-borne diseases, they were always here. The dogs were always suffering of anaplasmosis and ehrlichiosis, and the same for the cats. Yeah, so the fact that we speak about hepatozone in cats just in past few years, it doesn't mean that the infection was not here. It was just totally overlooked, totally unknown by practitioners. Yeah. So logically, more we focus on the wild carnivores like jackal on this picture, which is definitely one of the fast spreading animals, the wild animals in Europe. We cannot say it's invasive because this is a natural spread from the Near East across Balkan Peninsula through lowlands along Danube, reaching already also Italy and spreading further in Italy to the south, and also throughout Central Europe, spreading to the north. And once when we speak about the vector-borne diseases of domestic dogs, we should keep in mind that jackal, the European golden jackal, is quite close relative to our dogs. And compared to wolves, it commonly lives also closer to human settlements, and it really creates in the environment the source or potential source of infectious diseases that are then transmitted by ticks to the domestic dogs. Okay, so we also discussed during my last talk that pyroplasmids, Babesia, and Theileria in fact represent quite a complex group of pathogens. And even those that are associated with domestic carnivores, mainly dogs and cats, still it's not a homogeneous group, which explains also huge differences in biology, huge differences in the vectors, huge differences in the treatment also that we discuss later. If you look to major groups of pyroplasmids in our or in carnivores close to humans, I mean mainly dogs and cats, we can distinguish at least 3 major groups that belong to totally different parts of the spectrum or of the phylogenetic clade of pyroplasmids. And it's a so-called clade 6, which contains the typical Babesia, like Babesia canis, rossi, vogeli, and so on, gibsoni. Then it's clade 3, which is Citauxone, relatively unknown parasites of cats that seem are quite common in Europe. I'll speak about it later. And then clade that we call the down there by green color, I marked the clade that is called clade 1, which is the clade that even taxonomically is unsettled, and which explains why someone called these parasites Babesia. Some other researchers in all the studies, they call them Theileria. And to be very honest, it's neither Babesia nor Theileria. It's simply a genus that, or assemblage of species that requires reclassification. And from the spectrum of pathogens, we will speak today about Babesia vulpes, formerly called Theileria annae, belongs to this group, together with some other species. And so it's quite a diversity, and each of these groups, it's quite different from the others. Yeah. So let's start with Babesia in dogs. Canine babesiosis, it's a disease that is well known to veterinarians in many regions, also well known to dog owners, because it can be quite acute disease with quite prominent clinical features that are visible also to the dog owner, not only to veterinarians. Yeah, so there are at least 3 species involved. I just want to remind you, and now I speak about that yellow insert on the right side, that particularly in case of Babesia and Theileria of carnivores, the taxonomy really matters. During my talk, I'm trying to avoid the taxonomy as much as I can, because you are veterinarians, you are not taxonomists, you are not parasitologists per se, you are vets, or you will be vets, most of you. But here we should keep in mind that in this case, the taxonomy itself, it's complicating a bit the story of canine Babesia, because historically, all those 3 which are in yellow insert, as you see, were treated or were considered a subspecies of Babesia canis, which for normal clinician is just mean it's Babesia canis. Who cares about subspecies? Yeah, it's canis. However, nowadays we know that these 3 are not a subspecies. They are totally different parasites that are having different tick vectors and totally different distribution. And we can say that we can conclude that they truly cause 3 different diseases. So nowadays, when we speak about Babesia canis in Europe, we speak only or exclusively about the one that was previously called Babesia canis. Yeah, the first one on the yellow box. The others are considered Babesia vogeli, Babesia rossi, and this is also the treatment that I follow in my talk. So if you look to Babesia canis, transmitted by Dermacentor reticulatus, with possible wild carnivores as reservoirs. And it's a typical cause of quite acute hemolytic anemia in dogs. It belongs to so-called large babesia. Its microscopic detection is quite easy and can be easily performed on your desk if you are having microscope in your practice, if you will have microscope in your future clinical practice. And of course, the detection can be conferred by a range of different PCRs and real-time PCRs and so on. Even though we say that it's causing an acute disease, acute febrile disease in dogs, still, majority of cases in endemic areas are asymptomatic. But this is true for all babesia, and I really hope that you will keep this in mind. And when we speak about babesia tail area, it's a kind of iceberg. There is few animals on top which are clinically ill or can even die, but there are much more animals which are asymptomatic at the lower part of this pyramid, which usually doesn't require any attention of veterinarians. So anyway, when the disease is acute, it's causing fever, hemolytic anemia, which manifests, and this is what we shown already in the yesterday's talk, by so-called pigmenturia, or more specifically by hemoglobinuria. So hemoglobinuria is the first thing usually the owner observes, the same like we discuss in cattle, together with the fact that the dog is easy to it's tired, it's apathic, it's febrile. And if you look more to the, I would say, features detected within clinical pathology, we commonly see not only hemolytic anemia, but also it's commonly associated with thrombocytopenia. The rest of the features in the hematology is not really specific for the disease. Yeah. So it's large babesia, easy to recognize in the blood smears if you make a good blood smear. Later we speak about the diagnostics. Yeah, the most typical one on this picture is the one here in the middle at the lower part of the slide, where you see those two typical merozoites connected, because the parasite reproduces by binary fission. So in some of them, the younger forms can look like this one on the upper part of the slide, where we see only one, but then it divides into two, as we see here in the middle or here. So this is typical large babesia of dogs. Not that difficult to be detected in the blood smear. Here, sorry for the Czech language. Here, it's just a picture from our recent article about the emergence of canine babesiosis in the Czech Republic. I just want to show here, first of all, the erythrocytes on the right side that are absolutely not that clear as those that you see here, because simply the clinician in the clinic was unable to make a good blood smear, they had not done it properly, and so on. And this is the biggest issue of detection of babesia. You really need to play with that a little bit. You really need, or you have to have, or you need to have high quality blood smear, well working staining to be able to detect that. But the most important thing on this picture is right in the middle. You see two syringes that doesn't contain blood. They are quite dark, as you see. It's not a blood. It's just a urine from spontaneous mixture or urination. And this is why the owner came to the clinic. And this is what the owner observed. The dog was apparently ill and was having almost chocolate brown urine, which is sometimes not that easy to spot if you don't truly watch your dog urinating. In other words, it's good kind of message to convey to the dog owners that if they are living in the babesia endemic area where the infections are common, and if they don't or if they feel that something is wrong with their dog, they should watch its urine. They have to try their best to see it urinating pink, and they should at least visually control if the urine is normal color or if there is some change in its appearance. So hemoglobinuria in case of Babesia, quite important diagnostic feature. We all discussed that all around Europe there is ongoing discussion if wild carnivores can be a reservoir of this particular species of Babesia. And we still do not have answer. The answer probably is yes, occasionally, but perhaps, or maybe, but it's not common. In our recent study from Poland, we detected the infection in a few foxes in the area where the infection was much more common in dogs. And it seems that if there is a wild reservoir, it still doesn't play the true importance, and the infection is mainly shared among the dogs or theoretically between jackals and the dogs. So the foxes, which are the most common canine carnivore in Europe, they don't play significant role, which is big contrast to one of the few next Babesia that we will discuss. So in this case, maybe, but not important. Babesia vogeli, it's a kind of sister species of Babesia canis. I would say it's less pathogenic Mediterranean species of sister of Babesia canis. Formerly, it was called Babesia canis vogeli, as we discussed, and it's typical infection in Mediterranean area because of the distribution of its vector. It's also causing hemolytic anemia. It's also large Babesia, but it's more frequently asymptomatic than the previous one. And the infections are in endemic area are, I cannot say quite rare, but they are definitely not as common as Babesia canis in the new Central European foci. And also the clinical signs are usually not that prominent. Also, the treatment is identical to the previous one. We'll speak about that later. So this is what you probably have quite commonly around you in Sardinia. I don't know how frequently you see clinical canine babesiosis. I'm coming in spring for ticks to your beautiful island, so I truly look forward also discussing these things around with you or the vets around, because it's definitely interesting disease. The third species of Babesia, which can be found in European dogs and is found in the European dogs, it's different. It's considered being so-called small Babesia, not because of its taxonomic placement. It just really shows its size. So it's a bit more difficult to diagnose. The merozoids and other stages are truly small in the erythrocytes. It's also transmitted by brown dog ticks, which again are those that are common also around you in Sardinia. It's said that it's, compared to the others, it's more pathogenic also to puppies. There is evidence for transplacental transmission, and then the young dogs or small puppies can die relatively soon after they are born. And what's interesting, it's transmitted not only by tick bite, which is normal, but it's transmitted also by dog bite. It's difficult to say how frequent this is, but in any textbook, any chapter about Babesia gibsoni, you will read about the cases of serial transmission in fighting dogs, breeds specifically in the United States, where the dog fights were at least maybe hopefully were, but still are in some areas quite popular, even though not legal. Yeah, so it's a disease that in fighting dogs can be truly transmitted by bite. Not that surprising, because when the dogs are fighting, of course, they injure also their mucous membranes, and there is spontaneous bleeding. And it's not that surprising that the parasite, which is associated with erythrocytes, is transmitted by this way. Though it's not the dominant way, for sure. Yeah, ticks are necessary for the long maintenance of the infection. Yeah. What's important here is the last point that I want to stress. The rest is almost identical, but what's important is the last point. And this is that specifically in Southeast Asia, not Southeast, South and Southeast Asia, there are the strains which are totally resistant to atowacon azithromycin treatment, or specifically to atowacon, which is the most recommended treatment for Babesia gibsoni. Here in this species, we don't use the imidocarb. And what's interesting, we nowadays, because the allele that is the allele of the gene that is associated with this resistance was in the parasite was more or less accidentally identified in some studies years ago. We can test this by PCR. So if we have a case of Babesia gibsoni, before we start the treatment, we can, by a relatively simple PCR, test whether we can expect this strain to be resistant to atogacon or not. And it can, of course, further direct our treatment regimen. Because, of course, we don't want to, first of all, it's not that cheap and not that easy to get the drug. And if the dog is, or if the parasite is resistant anyway, then why to try? Then it's better to go the alternative way. So this is quite unusual among the pyroplasmids. In other Babesia, we also see the resistances, or we expect slow emergence of resistances, but we don't have the tests to detect them that fast as in this case. So that was Babesia gibsoni, the third species. And the fourth species of Babesia in dogs in Europe is quite specific because it's typical parasite of red foxes, only accidentally transmitted to dogs. And we already discussed this probably yesterday, that taxonomically this parasite was historically called Theileria annae. Sorry, there is a typo, there is R missing here. Theileria annae or Theileria microti-like. So if you look to the older literature, even with good recommendations for the treatment, you should also look for these parasites under its older name, Theileria annae. We don't know much about its occurrence in red foxes because no one truly wants or have tools and time and interest to study it in the red foxes. We only know that it's extremely common. Extremely common mean that, for example, in the Czech Republic, when we take different populations of foxes, the prevalence might be between 20 and 40%. In some localities, even more. So theoretically, almost every fox or every second fox is having this infection without any clinical features. On the other hand, to be honest, we don't know that much about the clinical things in foxes because who cares? Yeah. Maybe they suffer from some mild periodical anemia, but we cannot detect that, of course. Yeah. So other interesting thing with this parasite is that the tick vector is not yet fully identified. We assume that these are mainly the ticks of the genus Ixodes, but not clear yet. What's important that in dogs, it can be symptomatic. So it can again produce the anemia and microscopically positive cases when we see the small Babesia in the blood smears. And there is also a range of different PCRs that can support the diagnostics. From the cases that are known in the literature, it seems that the infection is more prone to develop, I mean clinical infection, to develop in immunocompromised dogs. Just very recently, like a couple of months ago, I see a dog here in the Czech Republic that was an old splenectomized dog after some, I don't know, it was probably injury, I don't remember, doesn't matter. So the splenectomia was performed. The dog was not really doing well, but was also living. It was old dog, but then the dog died because of Babesia vulpes infection, apparently after contact with some ticks in the environment. So it can be little, and if you go through the literature, there are well-described cases that occurred all around Europe in dogs. And as again, we know nothing about foxes. We just know that it's very common. So that was Babesia. If you look to Babesia in cats, there is one special thing, and it is that we call them by their own name. Citauxone, it's more or less also a small Babesia However, for this particle clade, for this particle group of Babesioas, we used the separate generic name historically, Citauxon. It's quite a difficult name even to pronounce. It's a very difficult name to write it, but it's good to learn that because it seems that it's not that rare in European cats that was believed until recently. And it's also not a single species. Until relatively recently, it was considered as a single species, Citauxon felis, but then some other species were described, and recently in Europe, 3 species were described based on slight genetic differences. We know nothing about if they differ, whether they differ in clinical features of the disease in cats. We don't know if they differ in vectors, because even for these parasites, we don't know exactly the vector. Again, it's probably Ixodes. And definitely it's something that deserves more attention. Yeah. So in the diagnostics, again, they appear as small Babesia in smears. They can cause acute or subacute infections in domestic cats. They can be totally asymptomatic in wild cats and lynx. And the clinical signs are not very specific. And also the hematological changes are not really specific enough to support significantly the diagnostics. So the diagnosis is usually based on a combination of clinical symptoms, detection of small Babesia in the bloodstream, in erythrocytes, and PCR confirmation of the infection. So let's discuss now a little bit more about the diagnostics. So a lot of was written about the diagnostics of Babesia in dogs and cats, and still almost weekly the questions from my colleagues in the veterinary field are emerging regarding the diagnostics, because it's still a tricky chapter in veterinary parasitology. First, before we start discussing how we diagnose or how we detect the babesiosis or Babesia, Theileria or whatever in dogs and cats, we should ask other question, and it is why we want to detect them. Because there is really huge difference whether you are a clinician who deals with an acute clinical case on his desk at his clinic, whether you are a pathologist that is trying to answer the question about the cause of mortality of given animal that you are again having on your desk, or whether you are a parasitologist or epidemiologist doing research and the surveillance for the parasite. Then the tools are different, and we should keep it in mind when reading the studies. So not everything what's good for epidemiology is good for the clinicians, and vice versa. So in other words, the different questions we ask require different methodologies to be used. So anyway, we have a cat. In this case, we can say, okay, it's a suspect cat. He doesn't look good, tired, apathetic, slightly febrile, decreased food intake. So can it be babesiosis or citauxonosis or not? Or going outside, of course, so time to time we are finding ticks on him. So how can I detect that? What can I do? Usually the diagnostic starts with a blood smear. Or my opinion, it should start with a blood smear. And I truly recommend to all clinicians around me, like, please do it. Do your own blood smears, examine your blood smears by yourself before you submit the case further, because this way you can have quite fast and reliable diagnostics. So, but how to examine it? What are the basic things they ask? In my opinion, there are 2 things to keep in mind, or maybe even 3, to answer how to do and how to examine the blood smear for presence of Babesia. Of course, you should find them when they are present. It means the blood smear should be good you should spend a significant time on examining it, going through all the available monolayer of cells to examine them. Not only to see the babesia when it is present, then it's usually easy. If you really find it, it's nice feeling. You see it, wow, this is it. Yeah, perfect, done. But you should do it good enough also to understand your negative result. And this is quite important. And this is the most difficult part. To trust yourself that nothing what you see in the smears is the result. Because in my opinion, it's useless, and this is what unfortunately a lot of vets are doing, to do a blood smear, to check it under the microscope just for a couple of, for a minute or two, and to say, oh, I'm not sure. Better to take the sample and send it to the lab. So if you are sending it to the lab anyway, then you don't need to spend time and effort for making the blood smear, of course. So you should trust your negative results. That also means you need some experience. I always recommend keep the positive slides so you can go back to them with the next cases, so you can kind of refresh how this parasite looks like and so on. And even when you detect it, and when you are sure, yes, this is a dog, yeah, or sorry, that was a cat. Yes, I see the small babesia in the blood. We still have to translate this finding into our own clinical praxis. And this is finally the most tricky part. We have to answer the question, fine, the cat has Cytauxone, but is this Cytauxone responsible for the disease that I see right now in this animal? And then what can help us is the quantity of the parasites, the general appearance of the blood cells, the other hematological parameters, and the clinical features. And we are guessing. If we don't truly see an anemia, if we see only a few parasites in the smear, we are still guessing whether this is the cause of infection or not. You can skip this. Yeah. Which I just say here. And we should always consider that specifically in dogs, not that much in cats, there always can be some diversity. So we cannot automatically expect, yes, this is dog, this is babesia, this is canine babesia canis, dot. No, it's not. Because in any part of Europe, virtually at any part, there are the foxes living. And if you have foxes around, then always you should consider, okay, is this large or small babesia? And if it is small babesia, is it Gibsoni or Vulpes? If it, yeah, and so on. So finally, usually we get, if we make a good smear, if we make a good diagnostics, if we spend time on that, we are somehow by microscopy obtaining nothing more, nothing less than this. The presence of merzoids in the erythrocytes. They can be big, they can be small, but we cannot truly identify the species based on the microscopy. Yeah. So then, logically, for the first hint, we know this is babesiosis. Then we can sequence the material, submit the samples to the laboratory and sequencing, getting the final confirmation, not only that this is babesia, this we already expect. If we see if we have a good smear and we see the parasites, we expect it's babesia. But we need to know which, or the sequencing can tell us to which clade of the babesia our pathogen that I see right now in my cat or dog belongs to. And maybe you can ask, or all of us can ask the question, do we really need to identify this species? Isn't it enough for me to say, oh, this is babesiosis? And the answer is definitely we need to identify them. Why we need that? We need that, first of all, I'm a researcher, I'm a scholar. Yeah, so we need to know that for the epidemiological purposes, we need to know what's around us. Even you need to know which parasites are present around the areas you live in Sardinia or somewhere else in Italy. So we definitely need this information. But we should keep in mind that there are the differences in clinical manifestation, there are differences in prognosis, and there are also differences in treatment. So to know the species as fast as possible can truly help you, specifically in the areas where babesioses are quite rare. If you see Babesia almost daily, as some of my colleagues nowadays, for example, in Poland or Slovakia, then you truly don't need to do so. Yeah. If you are a clinician somewhere around Bratislava in Slovakia or in Košice, and you have the sick dog in your clinic almost every week, then seeing the large Babesia in the smear together with clinical science is enough to say, yes, this is Babesia canis. Yes, let's start the imidocarb treatment. And in the absolute majority of cases, you are right. So there is quite nice review for those that want to learn more and understand more about the diagnostics of Babesia. There is a quite nice review in Parasites and Vectors that you can easily download and read. And if we look a little bit for the scheme, they basically summarized in a simple graphical scheme the procedure that we discussed until now. So let's follow it just to repeat this a little bit to remember that. So you start with a blood smear, where the blood smear is negative and you still want to know whether this can be or not the babesiosis, you submit a sample for PCR. If even PCR is negative, you can still consider the serology, which is then more the epidemiological than a clinical tool. Because if you don't see the parasite in the blood by PCR, if the PCR is done properly, and if it is the PCR that amplifies the parasite you are looking for among those three groups of Babesia, then we can say, no, the disease in this animal is not caused by Babesia, no Theileria. So the quantitative serology, I would say, is more, at least for me, more the epidemiological tool than the clinical tool. Yeah. Of course, if we see the Babesia, then you can check the size. It's the left side, a little bit left, or upper part of the chart, and then if you find it useful, you go through PCR again. So that's basically what we discussed. So let's look to the treatment only very briefly. We already discussed that there are three groups of Babesia or pyroplasmids in our carnivores. And we already discussed yesterday that specifically in herbivores, we typically use imidocarb dipropionate. So the question is, can we use it also in carnivores? Of course we can. It's still the most recommended drug for treatment of typical Babesia, specifically the large ones. Yeah, so the Babesia in clade 6, which is Babesia vogeli in Mediterranean, Babesia canis in more Central Europe, or if you are lucky enough and work as clinician in Africa, in South Africa, Babesia rossi is also considered sensitive to that. For the others, it's a little bit more tricky. For clade 3, which is Citauxone in cats, usually use Atovacon or Buparvacon combined with antibiotics, with azithromycin or clindamycin or clindamycin. And the same is true for Babesia gibsoni, which belongs to group 1. In cases when the strain of gibsoni that we have is resistant to Atovacon, then the combination of Atovacon clindamycin, specifically the high doses of clindamycin, can help, but it truly requires effort and monitoring of the animal. In all these cases, it's good to inform the dog owner or cat owner that it's more or less the clinical treatment that we are submitting, or we are giving the drug, not to clean the dog totally from the given parasite, but to help him to survive the acute phase of the infection. Why? Because it's quite common that we still see the presence of the parasite, specifically when we use the PCR detection, even after the successful treatment. So the dog looks perfectly healthy, is back to normal life, owner is happy, you are happy, invoices are paid, and then you do another PCR and the dog remains positive. Is it disappointing? No, it is not. That's normal. And if the dog is immunologically competent, if the dog is healthy enough, it doesn't require continuation of the treatment. Yeah. So again, I will direct you a little bit to the ESCAP guidelines, which are exactly for this purposes written. And I'm not mentioning the dosage in my slides because you can easily find dosage for those drugs in the charts and tables of the guideline number 5 of the ESCAP, which is already in its 4th edition. And as you'll see here on the slide, it's January 2023. So this is the most recent information about these parasites included there. So as a kind of homework or preparation for your exam, I truly recommend going into that. And the same if you have a case. I am doing the same. If I have if I see a case, I usually first consult the knowledge which is presented here with some more references, and then I recommend or not recommend the treatment. So that was Babesia theileria. Let's move faster because we still need to discuss hepatozone and some more parasites. So what is hepatozone? Why we speak about hepatozone together with the pyroplasmids? The answer is easy. Because in carnivores, it's also a tick-borne parasite causing a tick-borne disease called hepatozoonosis. Yeah. However, it's quite different in many aspects. And the first is that it doesn't belong to pyroplasmida. It's totally different group of apicomplexan. It is still apicomplexan, but belongs to so-called Adelorida, which is absolutely separated group of apicomplexan protists with quite different morphology, quite different biology, and quite different even ways of infections and so on. The infections are, again, quite commonly asymptomatic. I can guarantee you that if you examine the dogs around you, even though they look perfectly healthy and strong, you will find some hepatozone-positive animals among them. And in many animals, due to this, the parasite was overlooked for quite a long time, specifically in some regions and it's in some types of animals. And nowadays, there is a kind of revival of interest in hepatozone, and it's becoming almost a fashion to study and demonstrate the hepatozone in different types of wild living carnivores and so on, which is, of course, again, supported by the tools that, thanks to which we can detect the genetic diversity and keep describing new species and so on. Yeah. So what is hepatozone canis? Hepatozone canis is a protozoan parasite of white blood cells. This is a huge difference from Babesia. All the Babesia we discussed today in dogs and cats are associated with red blood cells, not hepatozone canis. It's a parasite of white blood cells. There are probably various tick species involved in the transmission. There is definitely a reservoir among wildlife, as we discussed already, and it has very variable pathogenicity. And from practical point of view, it's important to keep in mind that the infection is not transmitted from tick to dog by the tick bite, but by an ingestion of a tick, which is, of course, quite frequent because once the dog or cat are having the tick, they, of course, try to get rid of that. They bite it off, they chew it, they swallow it, they get infected. But then, due to this, we see some limits of the effect of the acaricides. Why? Because the dog can lick or can eat the tick, or cat specifically, which, cats are cleaning themselves very frequently. So simply they eat the tick even before the tick is in contact with the acaricide, which is present on surface of the host body or in its blood. So even well-protected dogs, like collars or pipettes, spot-ons or whatever, can get infected by hepatozone canis by simply ingestion of the tick. Yeah. If you look to the diversity in canines, we speak about two species. Hepatozone canis, it's common in the Old World, So it's the species living in Europe in our dogs. In United States or in North American continent, there is also another species, Hepatozone americanum, which has a bit different pathogenicity. It's more pathogenic. It's causing quite severe infections in dogs. It has significant part of the life cycle or stages occurs in the muscles, causing inflammations in there, and it's quite different and definitely more pathogenic. And then there are species in cats. Old species known for a long period of time from the beginning of previous century was Hepatozone felis. Very recently, other species was described first from wild cats in Europe. It's Hepatozone sylvestris, but nowadays we know that it's occurring also in domestic cats, not surprisingly, because it's the same species of cat, of course, the wild and domestic cats. So not surprising that they have the same species of hepatozone. How the hepatozone looks like, I already showed you before. So we can detect it in monophytes and neutrophils as a ovoidal or elongated gametocyte presented in the cytoplasm of the cell. So here you see very typical, here it is once again, very typical infected cell. So this is the nucleus of the white blood cell, this is the white blood cell itself, and the parasite is present in the cytoplasm. The problem is that it's not very frequently seen in the blood smears, even in clinically affected dogs, because also the white blood cells, to be very honest, are not that frequent in there as red blood cells. So sometimes it really needs effort to find hepatozone in a smear. And in many situations, we know, for example, by PCR, that given dog is positive, and we are, even though we examine a series of 10 slides, we are unable to find the parasite in them. So as I said, and these are the asymptomatic cases, which is by far a majority of cases. Yeah. On the other hand, as I already told you before, we also see the extreme deadly cases that are happening more frequently with Americanum, but also in Europe. We cannot really say that, no, it's not pathogenic, we don't need to care about Hepatozone canis. We should. But when we see the clinically ill dog with high parasitemia of hepatozone, we should also ask, okay, what's wrong with this dog? Because it's not normal. Normal for a dog is to stay asymptomatic. If it is having high parasitemia, it can also direct you a little bit to some immunity-related issues and so on. Anyway, not surprisingly, the disease is manifested by quite high fever, high neutrophilia, in some aspects by anemia, and also by hyperglobulinemia. Yeah, so these are the typical features of the infection. And what's complicated, and we discussed that already in other parasites, I don't go again and again, is the interpretation of the PCR diagnostics. Because PCR for Hepatozone canis is commonly included in multiplex panels for detection of tick-borne infection. So usually the companies or the professional or commercial labs, they are offering a kind of set of diagnostics for, they're offering to perform a set of diagnostics for tick-borne infections. And then you get the results for different tick-borne pathogens, including Hepatozone canis. And commonly the dog is negative for the others and positive for Hepatozone canis. And then the interpretation, what does this mean? It's quite tricky. And just to show that it's a reality, it's not that long time ago when we did some PCRs or when we were establishing our PCRs for Hepatozone canis in my lab, we needed a negative control. So for a negative control, you take any dog from a surrounding or around, and more or less you consider it negative. So I took just the dog that was sitting in next office in our vet school, that belonged to a colleague of mine. And I said, oh, can I take a drop of blood? We need a negative control to our PCRs. And even though the dog was perfectly healthy, it appeared as positive. So there are truly positive dogs all around. And they can be happy, they can be healthy, and they do not need a treatment. And this is, please, a message that I truly suggest you to take. Yes, do the diagnostics. Use even the the sensitive diagnostics for hepatosome canis, but then consider what to do with the results. This we already mentioned that historically the hepatosome canis was believed to be present in dogs mainly in southern Europe, being transmitted by Rhipicephalus sanguineus, the brown dog tick, which lives up to here. But recently we know that in whole Europe, the reservoir hosts are the red foxes, in which the parasite is very common. Very common mean, again, the majority of fox population can be infected. And what's still quite puzzling and not very easy to understand, I tried to explain that last previous week, is why we don't see the cases in Central Europe, why it is so rare in Central Europe, even though there are so many positive foxes, and why you see so many cases in Mediterranean, I mean the clinical cases. And this is still something that definitely deserves attention, and I cannot truly give you an answer, but it's for sure associated with the differences in epidemiology that are related to the fact that you have different tick involved in the transmission, which is Rhipicephalus. So when we speak about the hepatozone and the carnivores, we speak about a relatively small group of adelarids which are here. And it doesn't include only the infections in dogs and cats, but in fact, it's a group of different species and genotypes that affect different types of wildlife around us, including cats, including marten, including brown bears. There is hepatozone ursi, the parasite of brown bears, in Palearctic. There is a hepatozone present in squirrels as well, and so on. Yeah, so it's quite a broad assemblage of species. But, and this would be the topic of next slides, we should also speak a little bit about the others. And this takes us a little bit further from the carnivores. And we should speak about other vector-borne adelarids. Because as veterinarians, we can see them not only in carnivores. There is a group of heteroxenous apicomplexans that belongs to Adelorida. And that is, sorry, this is not supposed to be here. And it's quite diversified. And we call them by a group name hemogregarins. And they are typical parasites of poikilotherm hosts. They are typical parasites of fish, amphibians, and reptiles. And it's quite a diversified group. It includes also hepatozone, but it includes, for example, in lizards, includes the genus Cariolizus, all around Europe present in lizards. It includes Hemolivia in tortoises. It includes hemogregarins in tortoises and fish, some other species in fish. It includes also, this is not heteroxenous, but it includes also Klebsiella, which is in horses, but I'm not going to speak about. But we are today, I want to tell you something about the heteroxenous adelarids, which are so-called hemogregarins. Just to give you a few examples, just to know that it's not only dogs and cats that can suffer from this, or can be infected by these pathogens, but also the others. Yeah, and as you see already here, so these are the parasites in blood smears of amphibians and reptiles. Can I ask you, in which blood cells are these? We don't speak about canis, yeah, it's obviously a monocyte. But what are these cells which are infected? We are looking at pictures from reptiles and amphibians.
Red blood cells?
Red blood cells, exactly. This is the difference. In mammals, parasite is present in white blood cells, but in reptiles and amphibians, it's present in red blood cells. And I just want to stress that in red blood cells, you see the nucleus in these hosts. Also in birds, but birds, they do not have hepatozone and hemogregarins. Yeah, so it's just not be surprised that you recognize that quite well, that these are the red blood cells, even though nucleated. Yeah. If you look to the life cycles of these parasites, they are not that surprising. They always involve some blood-feeding vector, and they involve two ways of infection, possibly three. Commonly, and this is case of hemogregarins in tortoises, simply the tick is somehow ingested by the tortoise. We don't know whether it's accidental or intentional, but simply it is ingested, which makes it very similar to Hepatozone canis. In other hepatozone species, for example, in parasites of snakes, we know that other blood-feeding vectors are involved, like mosquitoes. There are a bunch of species transmitted by mosquitoes, but snakes are, of course, not eating mosquitoes. Then the parathenic hosts are involved, which can be reptiles or small mammals like insectivores, possibly also rodents. And then the mammalian host is infected, the snake host is infected by eating the parathenic hosts. Yeah. So if we can conclude what the hemogregarins are, we can say, and this would be nice if everyone remembers, these are the heteroxenose epicomplex parasites transmitted by blood-feeding vectors, typically in poikilothermic hosts. They are common in red blood cells. They are very easy to detect, as you see here. It's very easy to detect them because the red blood cell itself is very, very large in reptiles and amphibians. And the parasites are even larger. So you cannot overlook that in the blood smear. Yeah. They are in erythrocytes, and they are typical, of course, in reptiles and amphibians that are originating from nature. We cannot see them in the animals which are born in captivity, normally, indoor, I mean. Why? Because we don't have the vectors, of course, so they cannot be further transmitted, which is good and for some cases of investigation of the origin of the reptiles. And this is something I did as a service repeatedly for the customs in Europe, like where we were asked, can you tell whether this animal was collected in the wild or whether this particle, tortoise or snake, originates from captivity? So then based on the parasites, we can. So if, for example, the land tortoises, which are declared in Europe as captive born, if they do have Hemolivia mauritanicum in their blood cells, it's quite highly suggestive for the fact that they are originating, in fact, from wild, probably illegally collected in the wild instead of being born in captivity. So this can help. The major genera in reptiles and amphibians includes at least these four: Hemolivia, typically in tortoises, Hepatozone, snakes, lizards, Cariolizos, lizards, Hemogregarina, the aquatic turtles, and some other. We don't need to go to details. Yeah. So this is the case of the one that is common in Mediterranean in tortoises, Hemolivia mauritanica, transmitted by specific vector, which is Hyeloma egyptium, the tick, which is specialized for feeding on Mediterranean tortoises, mainly in Balkan and North Africa. And this is how the parasite looks like in the smear, so you cannot overlook that. Yeah, typical gametocyte at the periphery of infected erythrocyte. This is a similar situation in a snake. Again, massive infection, almost you see more than half of the erythrocytes on this picture are infected, but still, and this the second thing I want you to keep in mind about hemogregarins, their pathogenicity in reptiles and amphibians is extremely low. So even the snakes that are having half of the erythrocytes infected, they are still clinically healthy and doesn't require any treatment. The infections are quite commonly lifelong, so they persist for years, which shows that the parasite is somehow slowly reproducing. But somehow the reptiles, specifically in captivity, they are able to handle it, and probably the infected erythrocytes are still capable of transporting the oxygen. So we truly don't see the anemia, we don't see hypoxia and so on in these animals. And the life cycle is probably very slow, and the breeding process is reproduction process of these parasites is so slow that simply they are not destroying massively the erythrocytes, and the animals are quite okay with that. Yeah, so this should be considered. And I can say that almost never. The treatment is never recommendable in these animals. And anyway, it's not really well known how to treat it properly. There were some attempts with some antibiotics and so on, and Atovacon, but it doesn't really work well, and I truly don't recommend that. The only thing is that it can be quite difficult to communicate it to the owner. You have an owner or a snake, I don't know, emerald boa from South America. It's a snake that costs between 1 and 2,000 euro. The owner is coming to your veterinary practice asking you, please, can you examine that for the parasites? And you say, yes, sure, we can do flotation of the feces and we can do the blood smear. Then boom, you do a blood smear and you see it full of parasites. And then it's quite tricky and it requires some effort to explain the owner that even though the parasite, the snake, which is quite expensive, has a lot of blood parasites, it still doesn't need any treatment. So this I told you. And I want to, but this belongs to other thing here. Sorry. I want to, when we speak about the blood parasites and pet animals and exotic pet animals, I should in last 5 minutes or so speak about some other disease, and it's malaria. All of you, you know, you are living close to Africa, so you must know that malaria is a typical infectious disease of humans, of course, being transmitted by Anopheles mosquitoes or like around the tropics in all tropical continents and so on. However, it's a little bit least known that malaria parasites are present also in animals. And surprisingly, they are not typically present in mammals, even though some of them can be. But they are typical for other groups of hosts and these other birds. And for some of them, this can be quite pathogenic. I selected for my talk only 2-3 examples of blood parasites that are really pathogenic and blood parasite veterinarians truly have to cope with or deal with. As you see, for example, from this just randomly taken picture from web. So yes, for example, for penguins, avian malaria is truly a deadly disease that every veterinarian in a zoo having penguins can confirm. If you look to avian malaria a little bit in broader scope, we can conclude that this is called the disease, which is called avian malaria, is caused by 3 groups of Hemosporidia. It's the genus Leucocytoson, Hemoproteus, and Plasmodium. We can distinguish them in the blood smears. And within each of these genera, there is huge diversity of species that we cannot distinguish normally in the blood smears. They are infecting various avian hosts, commonly also having low host specificity and also low pathogenicity, specifically in the free-ranging birds. If you go today out of your campus and collect some birds around, I guarantee you that you will detect plenty of them with some type of avian malaria, and they'll be clinically healthy. However, in some particular situations, in captivity, in exotic or non-European birds or non-native birds, we see the resulting disease, which we call clinical avian malaria. And then we can find the parasite even in quite high numbers in the red blood cells, as you see here, those 2 parasitized cells in a bird of prey. Why penguins? It's truly a problem for penguins in captivity, and it's one of the main obstacles for even keeping the penguins in outdoor enclosures in the zoological gardens. And we already know that there is a couple of species of Plasmodium involved. And for all of them, the penguins are more or less accidental hosts infected by mosquito bite or by bite of mosquitoes that transmit the malaria from the free-ranging birds in all around us. So in every zoological garden, of course, we have the population of free-ranging birds, the local native free-ranging birds. We have, of course, the mosquitoes. And once you show the penguins outdoor, of course, you cannot protect them during the night from mosquitoes, and sooner or later the transmission is happening. And why particularly penguins are so sensitive for malaria, we can only guess. But definitely one of the answers is that they originate not only in their evolution, but also physically, like the species that are existing or they are kept in the zoos, they originate in quite extreme environment where simply the mosquitoes are not occurring and when simply malaria is not occurring. So they are not prepared for this because they evolved in the environment free of malaria. And they are then, not surprisingly, more sensitive than the local birds in European or American fauna. But speaking about the rare and expensive birds, penguins are not the only penguins, not only birds that die because of malaria. The same way as for birds from Antarctic regions like penguins are, malaria is highly pathogenic also for the Arctic birds, typically for birds of prey. And again, one of the most typical, one of the most common owls or raptorial birds in captivity nowadays in the zoological gardens, it's a snowy owl. And it's again a species which is quite sensitive to malaria infection or avian malaria. And in many zoological gardens, it's one of the major troubles or problems for the breeding of these birds, because specifically the young animals on the nests, they keep dying because of avian malaria. Again, transmitted the same way as we discussed by blood-feeding arthropods from the wild birds that are living around. And the same is true for the garfalcon, which is another Arctic species of falconid bird, in this case, very sensitive. Here we see the distribution of both of these species. Again, if you look at that, they live really in the far north. Yes, there are the mosquitoes there for sure, but apparently there is no avian malaria around them. So again, they don't have innate mechanisms or inherited innate mechanisms to cope with the infection, and they are more sensitive than the others. Yeah, so if you wish to read something more about that, another article for the homework about the clinical infection of in snowy owl. And when speaking about the atypical blood parasites in exotic birds, the last parasite, no, almost last parasite I want to mention, we still have like 7-8 minutes, is a disease called atoxoplasmosis, which is basically a visceral coccidiosis caused by isosporum parasites. So here we speak about the true coccidia in birds, but if you look at the smears, they are causing the infection of the cells in the bloodstream. It's called atoxoplasmosis. It has nothing to do with toxoplasmosis. And until relatively recently, it was believed that these infections in exotic birds, like the canaries also, are caused by separate genus of epicomplexans called atoxoplasma. Nowadays, we know that nothing like that exists, and that in fact, those are the isosporum parasites, the typical isosporum parasites, or more or less, but with atypical localization in parenchymatous organs of their only part of their life cycle. So these are the species of coccidia that are having like two more or less separated parts of the life cycle. The one in the parenchymatous organs, including the parasitism in red blood cells, and the intestinal part of the life cycle, including typical gammagone in the intestine and production of deoxys to the environment. Yeah. So during that merogonial part, typically liver, spleen, and are involved, and the birds can truly die, and they really keep dying for clinical atoxoplasmosis in captivity. So it's highly pathogenic, specifically for juveniles, and in many settings, it truly complicates the breeding of the birds, including really rare birds. The atoxoplasmosis, its life cycle and mainly its treatment was understood well based on the example of this bird, which I'm showing you the picture, which is so-called Bali myna. It's a bird related to starlings that, as the name says, occurred on the Bali island in Indonesia. Every one of you know Bali. The bird got extinct in the wild, but still was persisting in the 80s, 90s, was persisting in captive groups in zoological gardens. And there was, of course, a huge attempt or huge effort to save the bird from extinction, to breed these populations, and to reintroduce them back soon or later back to Indonesia. But the major obstacle for their breeding was etoxoplasma, which is called etoxoplasma rothschildi. In that time was called etoxoplasma. Now we know it's Isospora rothschildi. That was significantly contributing to the mortality. And until nowadays, when we want to keep these birds successfully in zoological gardens, for example, we truly need to treat them against this infection during the breeding period, or just before that and during the breeding period. Using the common anticoccidial drugs, typical is toltrazuril, given usually in water to the birds for a period of several weeks to keep them more or less coccidia-free, to prevent the transmission to their offsprings. And this works. So after that was understood, after the toltrazuril treatment protocols were established, the birds starting to breed quite easily in captivity, and there are thousands of them around. But still, we need to consider this pathogen because all breeding flocks are infected by this Isosporum coccidium, and it's a true issue. And the very last pathogen is a bit unusual. I will speak about it only very briefly, and it's also a helminth transmitted by ticks. When we speak about the tick-borne infections, we commonly associate them with viruses or with bacteria or with protozoans, but there are also a few helminths transmitted by ticks. And these helminths belong among the filaria, and they are part of one's quite specific or specialized genus called Cercopitaphilaria. And it's another typically or more or less Mediterranean type of parasite. Why? Because it's typically transmitted by brown dog tick. So we are back to the same story as with pyroplasmids and hepatozone. Yes, it's common in some particular part of Europe because the brown dog tick is present in there. And there are several species, at least 3 of them are recognized in populations of domestic carnivores. And what's interesting, they are usually not pathogenic, not at all. And they are quite unusual the way they parasitize the dogs, because they are in subcutaneous tissue, that's fine, but also their larvae are in this tissue. So they are not, the larvae, the macrofilaria, are not entering the blood smear, the bloodstream. So we cannot detect them in blood smears. Yeah, so normal blood samples are almost useless for detection of Cercopitaphilaria. And if you want to detect this pathogen in dogs, you need to do the biopsies, relatively large volume biopsies of the skin, which is quite a painful procedure for the dog, I have to say, because I was doing that a couple of times, and it's not really nice to make a hole to the skin of your dog. Then, sorry for check here. In these bioptic samples, you can detect the microfilaria, but just by simple maceration in basic saline solution in the tubes. So you put the sample into the basic saline, and the larvae are coming out, and then in the sediment, after you remove the piece of the skin, you can detect them by microscopy. So that's a typical parasite which looks important for parasitologists. I can talk hours about Cercopitaphilaria in dogs and other animals in Mediterranean. But for the normal veterinarians, the importance of these parasites, really minimal because simply it's not causing an issue to the dog normally. But it can be quite surprising finding in some of the biopsies, and also can be surprising postmortem finding. And they are transmitted by ticks. This is why I speak about them today, even though they are not protozoans. And that's it. So, before we go for lunch, because everyone is hungry, or at least me, I am, do you have any questions?
Seems we are all dead.
All that, starving. Okay, so, fine. So thanks a lot.
I
And
had one question, if I
tell me
can.
for sure, Barbara.
Thank you. If I may. First of all, thank you so much for your presentation, very clear. And I had a case that I was managing last summer of paroplasmid in sheep. And these sheep were symptomatic, but very generally. They had hypochromic anemia. And we isolated first with a blood smear and then with the PCR amplifying the 18S RNA, Babesia / microti, or
that's interesting.
And we couldn't really say why we found this paroplasmid in sheep. So I was wondering if you...
I never heard about it, to be honest, and it's interesting. How long fragment you sequenced? Like whole 18S or?
I think it was the whole fragment, but I should ask.
Well, definitely, it's interesting, and I would definitely suggest going for that and try to do something with that. At least publish that, because as far as I know, it's unknown. And for me, there are only two explanations: either it's unusual infection of sheep by Theileria annae, or not Theileria annae, but like Babesia vulpes as aberrant host, or there can be some related species circulating among the herbivores in Mediterranean, because if you go to literature, there are some so-called Theileria described from roe deer. And I'm not really sure if they are sequenced. And if so, this parasite can be just, I don't know what was the similarity to Theileria annae in your material, but this can be something different. So I suggest going deeper in that, sequencing more markers, COX-1, ITS, and trying to answer that, because for me that's really fascinating. Yeah, because we work a lot on deer and roe deer in Europe and so on. And if you have the material, if you are interested, I can connect you to my colleague Christina, who did quite nice molecular work on herbivores and pyroplasmids. And at least she can confirm whether this is really true Babesia vulpes. We have to call it nowadays Babesia vulpes, probably because it's broadly accepted name, or whether it's just something close to that. And that's interesting for sure. And it's, now for me, that's just really a fascinating case, because I never heard about this group of parasites in European herbivores. So go for that and stay in touch.
Okay.
We can sample more and do something for that, because when it is on sheep, you also have a chance to find even the life cycle, because it should still probably be persistent in there. So this sheep, they should have some ticks, of course, and so on. So this is, for me, this is really nice case. So
we tried to collect some ticks, but we couldn't find any because the owner already treated the sheep. But we are still in touch with the farmer. So
interesting. So please, if you have no further plans with that, we can really discuss and we can do something on that, because it's an interesting case for sure.
Okay, thank you. Really nice. Yes. Maybe I can type the email.
Yeah, nice. Perfect. Looking forward.
Ok, ci sono altre domande? Any other question? Seems no. So many thanks, David, and hope to see you here soon, sure. Maybe in spring 24.
Definitely.
And keep in touch.
Definitely. Bye bye. Buon appetito.
Ciao, ciao, ciao.